Hi, I'm Chloe. I'm a PhD student here in the UK at the University of Liverpool. And my PhD question is focusing on how we can understand environmental signals and their propagation through a sediment routing system, which can be replicated by a granular avalanching system. So just to start off with some sediment routing. So sediment routing systems are thought of as constantly supplying sediment to be transported from source to sink. But changes in environmental boundary conditions, so for example, climatic shifts or a tectonic uplift, can generate environmental signals in the erosional engine. So when I refer to an environmental signal, I'm referring to a measurable change in the volume of sediment produced and transported down a system as a result in a change in one of these conditions. So when a signal is input to the system, such as on the diagram, naturally this should propagate down system to then become deposited in the sink. However, in this system, we have autogenic processes operating internally within the routing system, and this can interrupt signal propagation and cause unfaithful transmission due to temporary sediment storage. So therefore, an input signal of a certain period of magnitude can be buffered, modified, or even shredded as it passes through the system so that the final output at the sink doesn't actually resemble the input condition that was input to the system. So why should we study this? So understanding how signals propagate across a landscape to become preserved in stratigraphy is of key importance when we're trying to understand landscape response. Understanding which signals will be preserved in stratigraphy helps us to quantify landscape resilience, reconstruct past conditions and events, but also it's really important for understanding how landscapes will respond to future changes, whether these are natural or anthropogenic. So to understand signal propagation and shredding, I want to focus on some previous work in the field, which is generally focused on which signals will make it through the shredder to be evident in the system output, using experiments and numerical models. But all of these have tended to focus on the surface of experiments or across the top of a landscape. So all the graphs you can see on the slide here have a y-axis showing some form of flux measurement and an x-axis of time. And all this work has focused on propagation across a landscape, which has been really useful to provide a base into which periods and magnitudes of signals may be preserved or shredded, and to generate some really key timescales. But to be applicable in this field, we now need to focus on whether a signal is preserved stratigraphically. So the record, therefore, can be used to analyse the effect of external perturbations on a sediment routing system. So as I showed in the last slide, this previous work has focused on surface signal propagation in terms of which signals make it through and the thresholds for this. However, what we don't currently know is exactly how a signal propagates, or how it is diffused or shredded along its trajectory. Alongside this, if a signal was successfully transmitted across the surface, how does this then become preserved in stratigraphy? So this highlights a really key theoretical gap between signal preservation in the surface and subsurface, which I hope to look into. So to understand this, we now need to focus on processes internal to sediment routing systems and ask a large general question of exactly how do these periodic sedimentary signals make it through to the sedimentary archive. So we must develop the theory that I showed earlier. We must develop this further by linking autogenic and allogenic processes, as the rate and scale of autogenic processes determines the ability of an environmental signal to propagate and therefore be preserved. So to provide insight into this large, more general question I've given, I want to try and answer a series of smaller questions. So these include the first one, so how do signals become shredded as they propagate? Second, how does imposing an allogenic forcing onto a sediment routing system affect its internal dynamics? And thirdly, can we find a relationship between some of the key timescales which have been defined in the literature? So for example, Tx and Tc, which I will explain in more detail later. So as mentioned previously, processes internal to sediment routing systems, autogenic processes, are of fundamental importance when we're trying to look at signal propagation, but previous work has mainly focused on allogenics. So these autogenic processes, so for example, such as self-organisation of bed forms or channel networks, operate stochastically, and these cause transport systems to operate in quite narrow corridors, so that wide areas of a landscape are in hiatus at one time. So I've got a video on the left of this slide, which shows an experimental delta system. And as you can see, the channels occupy quite a small area of the delta plane, but as it avulses through time, this allows areas of activity and areas of stasis to migrate. So this promotes temporal sediment storage and release. And as you can see, this constantly changes how the landscape looks, and the resultant sediment transport configuration and consequently this shapes the stratigraphic record. So these processes are inherent to sedimentary systems, but they also set the lower threshold for the propagation of sediment flux signals through a sediment reaching system to the stratigraphic record. So for example, if we look at the diagram on the right of this screen, this shows how an incoming sediment flux signal is partitioned into a maintenance flux, which you can see by the grey arrow, an autogenic flux, seen by the green arrow, and bypass, seen in the darker grey arrow. This promotes sediment storage and transfer. So over the next couple of slides, I want to highlight two real key studies which forms the background to our research questions. So firstly, I want to look at Jerolmack and Paola 2010, who focused on signal propagation across the surface or across the landscape. And to do this, they've used a 1D avalanching rice pile, which you can see on the left. So the rice pile undergoes a series of storage and release events, so it has its own internal processes. These avalanches equate to the storage and release of sediment, which we could have seen on the delta plane on the previous slide. So using this system, they've shown that even under a constant grain input, the system output shows the fluctuations over a wide range of scales because a variety of avalanche sizes actually come out of the system. From this, they've generated this timescale Tx, which can be seen by the dashed line on the power spectra, which is where the variability saturates, and this marks the timescale between allogenic and autogenics. And this scales, as you can see in the box on the bottom, as the lengths of the system squared divided by the input rate. So using this timescale, they imposed a variety of signals onto the system, which you can see in the left-hand column of the right-hand diagram. And they identified that signals with a period greater than Tx were preserved, and this can be seen as a spike in the power spectra. But they also defined this magnitude threshold, where even if the signal had a period less than Tx, it could make it through the shredder because the magnitude of it was sufficiently large to overwhelm the system, as you can see in the two red circles on the screen. So this shows how the rice pile can take an input signal and shred it over the range of autogenic scales so that the signal can be completely destroyed. So secondly, I want to look at Tobial 2019, who built on the work of Jerolmack and Paola, but he also applied it to stratigraphy, where they look for evidence of different signals within the stratigraphy of an experimental delta system, as you can see in the left-hand image. So using this system, they imposed 4 different signal types, which you can see on the right-hand diagram by the ticks and crosses. And they analysed for the presence of these using volume change across the delta and through stratigraphy to see evidence of sediment storage and release. So they chose to identify the signals based on TC, which is the compensation timescale, which is defined in the bottom box. And this is thought to equate to Tx in the rice pile, but this scales as the maximum channel depth of a system divided by the aggregation rate. So we can here on the right-hand diagram on the left-hand side of this, we can see the time-dependent magnitude threshold, which they quantified, which allows the differentiation of surface and stratigraphic signal preservation, as well as shredding. So by the ticks and the crosses on that diagram, we can see that 2 out of the 4 signals imposed were preserved in the stratigraphy, whereas the signals with the shorter periods were actually shredded. So let's focus on my project. So to allow us to focus on the autogenics of the sediment routing system, we adopted a similar methodology to that of Jerolmack and Paola, and we opted to use an avalanching rice pile. So the work of Jerolmack and Paola shows how adaptable the rice pile is to be applied to analyse signal shredding at the system outlet. However, previous work on the rice pile, for example, Fret et al., has shown the ability of this to analyse the internal dynamics of the system, which equates to the autogenic processes present in the sediment routing system. So, a bit of background to the rice pile acts as a whole sedimentary system, where we can put an input rate of sediment in, we can analyse transfer and storage of sediment, so we can see rice buildups and then avalanches, and then we can also look at the output of this system. But this is great because the rice pile is a really simplified system, which allows us to explore processes which are present in natural systems that we can't yet explore in the field, and it allows us to understand fundamental dynamics in a really simplified way. So originally, we wanted to use the experimental setup of Jerolmack and Paola and actually physically build a 1D rice pile over at Tulane University. But due to the pandemic, we've now adapted this. We've built the rice pile as a discrete element model using MFIX, which is an open source multi-phase flow modeling code written in Fortran and built by the National Energy Technology Laboratory. So the DEM model allows individual particles of the solid phase to be modelled whilst the fluid is then treated as a continuum. So I can play you the video. There we go. The model was built to the same dimensions as Jerolmack and Paola, with 2 square sides which were spaced 2 centimetres apart. And the left-hand side of the container is closed, and the grains are inputted at a specified yet constant rate through an inlet. And then the right-hand side of the container is left open, which allows the grains to leave the system during the run. So when the model is run, if I play this video, the grains enter the model at the specified input rate, and the system undergoes a series of storage and release events, such as like the real rice pile would, which allows us to look at propagation of grains through the system. So here in that video, you could have seen a brief time period in the model run, but there's also a still image panel on the right-hand side. And we can identify the active layer of grains at any time period during this run by the velocity profile of the output. So during the run, MFIX assigns each grain in the system with a particle ID, which allows the grains to be tracked. So here we can see the data from a short model run, where the movement of particle 3469 is highlighted by the red circle. It's actually pink, but I've tried to make this easier to see. So this can be done for every particle in the system over every time step. And MFIX uses a coordinate axis system to define the X, Y, and Z coordinates alongside the velocity, which you can see in the spreadsheet on the right-hand side. And using this data, we can track the width of the active layer, the trajectories of individual and clusters of grains within the rice pile, but also start to look at length scales of propagation, residence times, and internal dynamics to a really quite high level of detail. So the major limitation of MFIX is actually that the grains are spherical, whereas the rice is elongated. But the elongated nature of the rice is actually really important, as it allows it to interlock, as you can see on that image on the left-hand side from Fret et al. And this generates the self-organized nature of the system, which is seen in nature. The MFIX can't currently change the shape of the grains, but what we can do is change 2 key parameters, and these are the friction coefficient and the coefficient of restitution. And these influence grain interactions, the angle of repose, and the mass flow rate. So to ensure that the parameters are correctly assigned, we're going to systematically vary the friction coefficient and the coefficient of restitution over different model runs in a matrix and compare this to real-life experimental data, so we can quantify which combination of values best represents the results of the physical experiments. But alongside for doing this for calibration purposes, this is also great because we can analyse the effect of these parameters on autogenic activity within the model. So just to show you some brief results, we've currently done 1 model run, basically to see how the model works and what the output data looks like. As this is
2 minutes, Chloe, 2 minutes.
No problem. And this has taken quite a long time to set up. So here we can see mass efflux on the left-hand side, showing that under a constant rate of 14 grains a second, the output fluctuations show over a range of scales. And then the power spectra of this seen on the right-hand side, which shows an approximate red noise growth up to a peak at the top before it decreases over increasing observation times described as blue noise. And what this means within the rice pile is that over short timescales, a high flux is generally followed by another high flux. But if we look at this over a longer timescale, the high flux generally means a larger failure events occurred. And for the system to properly regenerate, the fluxes will need to be small at the next time step. But if we quickly compare this to that of other experimental data, we see different power spectral trends appear. So the data from Straub follows a very similar pattern, whereas the Jerolmack and Paola data shows a red noise changing to a white noise pattern. But from our calibration, changing the friction coefficient and the coefficient of restitution may lead to changes in the power spectra, which might provide more insight into the internal dynamics of the system, which shows the need for the calibration. So just to quickly conclude, so far, the MFIX model does show promise in its ability to replicate the results of the rice pile, but this now needs to be tested further and calibrated properly to see whether it undergoes the same internal dynamics. Once the calibration has been achieved, we hope to use this to investigate the effect of autogenic processes further by tracking particles through the system. So overall, we hope this work will currently advance the understanding of signal shredding, focusing on autogenics rather than allogenics, to provide information on how signals propagate and how the shredding process occurs. Great, thank you so much for listening, and I'll take any questions.
Okay,
Brilliant,
thank you so much.
Chloe.
Yeah, that was great.
Go for it, Emma.
Yeah, so we're open to questions. So either, you know, write it in the question chat box or raise your hand. So we've got a couple of minutes.
While we're waiting, I have a question for you, Chloe. Are you planning also to vary the width of your virtual box or in your model?
Currently, we're using the dimensions seen in the Jerolmack and Paola paper purely because we have the results of that, and we can. I mean, fingers crossed. We're currently setting up a rice pile in Liverpool as we speak, which we can set to Jerolmack and Paola, and then we've got the data to compare to. But if we see that that actually works, then and we know the internal dynamics are replicated, then definitely we can vary the width and vary the length to see how this influences it.
Great. So I have actually another one. So
Sure.
yeah. So do you think that the the signal you see in your rice result, to which part of the source to sink environment you think that would reflect the, you know, be be a parallel to it, the most accurate? Does it make sense? That was terrible English. I'm sorry.
Do you mean which part of the system would shred the signal?
Or the signal, the shredded signal you see in your rice experiment. Which part of the source to sink system do you think your rice experiment will be the closest to the reality? Do you know what I mean? If it makes sense.
Sorry, I'm understanding you now. So the rice pile has to be thought of as basically a whole system. Where the inlet where you input the grains is basically the right up in the mountains, and then the outlet is thought to be ocean,
The deep sea.
and then all the rest of it is just a transport system. But we're hoping that in the calibration of this, if we can change the two parameters alongside maybe the type of grains or different things within it, we might be able to find a link between different types of environment and different shredding. But then that'd have to be carefully thought out about which parameters convey a certain environment, if that makes sense.
Yeah, totally. No, this is great.
We thought
super
about
cool,
that.
super
It's just
interesting.
a work in progress.
Yeah, no, super cool.